Hi guys, welcome to Medicinal Chemistry 1. This lecture will be on neuromuscular junction blockers, and my name is April Johnson. So I will take you through, just a general introduction of what we're doing, and then I'm going to go into neuromuscular junction blockers, that is the drugs that affect this action. And then I'm going to mention briefly the drugs affecting the autonomic ganglia, and then I have some questions interspersed throughout the lecture. But first, let's listen to this video.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the divisions of the nervous system. There are two major divisions of the nervous system. The first is the central nervous system, which is made up of the brain and the spinal cord. The second is the peripheral nervous system, which consists of nerves that run throughout the body. The peripheral nervous system itself is made up of two subdivisions. The first is the somatic nervous system, which contains nerves that carry sensory signals from the body to the central nervous system, and nerves that carry motor signals from the central nervous system to the skeletal muscles. The somatic nervous system is associated with voluntary movement. When you clicked on this video to play it, the signal to depress your finger was sent from your brain to your finger via the somatic nervous system. The second division of the peripheral nervous system is the autonomic nervous system. The autonomic nervous system is sometimes called the involuntary nervous system, and it is involved in regulating the internal environment of the body. It carries signals from internal organs to the central nervous system, and from the central nervous system to the internal organs. In this way, it is involved in regulating things like digestion and heartbeat, which are generally outside the realm of conscious control. The autonomic nervous system can be further subdivided into sympathetic and parasympathetic nerve fibers. The sympathetic nervous system plays a large role in stimulating and mobilizing energy resources, while the parasympathetic nervous system acts to conserve energy. For example, if you're in a frightening situation, the sympathetic nervous system will cause your heart rate to increase, your blood pressure to increase, and your sweat glands to be stimulated. If you're eating a meal, however, and are not frightened, your parasympathetic nervous system will stimulate digestion, increase salivation, and slow your heart rate. Due to these functions, the sympathetic nervous system is often described as being involved in fight or flight responses, while the parasympathetic nervous system is described as being involved in rest and digest responses.
So just to reinforce what you saw in the video just now, that we have the autonomic nervous system and we also have the somatic nervous system. So what we need to know is that for one, somatic, there's no ganglia, while there is for the other, right? And we're also looking to distinguish between the sites of action of these drugs. So for the ganglionic blockers, they act here, right? As the name suggests, ganglionic blockers. And then there are the neuromuscular blockers, and this is their site of action, right? So again, we have to distinguish between the autonomic and the somatic nervous system, right? And then, that is ganglia versus no ganglia. And then for the sympathetic and the parasympathetic, we're looking at the transmitter, right? Acetylcholine for the parasympathetic only. And then for the sympathetic, we do have norepinephrine, right? And epinephrine. So just to point out these things. So again, just to reinforce, for the autonomic nervous system, we have the preganglionic neuron, then we have our ganglionic transmitter, and then we have our neuroeffector transmitter. Okay, so those are the differences between the sympathetic and the parasympathetic nervous system, as well as the autonomic versus the somatic nervous system. So this leads us now to question 1, which is, what is the difference between the somatic and the autonomic nervous system? And the answer is the somatic nervous system is a component of the peripheral nervous system associated with the voluntary control of body movements via the use of skeletal muscles. And as opposed to the autonomic nervous system, which is a part of the nervous system that supplies the internal organs. So, for example, the heart, the sweat, the salivary, and the digestive glands, this system works automatically. So our follow-up question 1, so all of the follow-up questions, I expect that you will have responses to them at our next class. Okay, so this is our first follow-up question. What is the difference between the parasympathetic and the sympathetic nervous system? So just pause the video and answer. Well, you don't have to pause the video. For the other questions, you should pause it and then look at the answer. But for these, you can, well, you decide how you want to do it. But these answers should be presented at our next meeting. So as I mentioned before, we're looking at neuromuscular blocking drugs, right? So these are muscle relaxants, and they block the cholinergic transmission between motor nerve endings and the nicotinic receptors on the neuromuscular end plates of the skeletal muscle. So this is a picture of a neuromuscular junction. So as the name suggests, the neuron and the muscle, right, at the end, that's where they meet. So in the space here between them, we refer to this as a synaptic cleft. And so if our direction of movement is from the nerve to the muscle, then this is referred to as the presynaptic terminal membrane, and then this is the postsynaptic region, right? So this is our muscle, and we have our receptors on the muscle, and they're referred to as nicotinic cholinergic receptors, right? So our acetylcholine is transmitted from our storage vesicles to our receptors, and then we have the resulting action. So what we must know here is that we must know the structure of acetylcholine, right? And it's abbreviated ACH at points throughout this lecture. So we must know it's a simple molecule. We must know the structure. So here we're looking at the binding of acetylcholine to receptors on muscle end plates. So in this, this dark square, we have our acetylcholine and we have our nicotinic cholinergic receptor, which looks almost like a lock and key scenario, which once the acetylcholine interacts with our receptor, our ion channel, which is closed here, is now opened, and then we have potassium exiting and sodium and calcium ions. Notice they're all ions, right? So sodium and calcium ions entering and potassium ion leaving. All right, so as I mentioned before, today we'll be looking at muscle relaxants, and particularly depolarizing muscle relaxants and non-depolarizing muscle relaxants. And for the depolarizing group, we'll be looking specifically at succinylcholine, otherwise called Suxamethonium, and we'll also be looking at Becamethonium. And for the non-depolarizing muscle relaxants, we'll be looking at several that fall under the categories of short-acting, intermediate-acting, and long-acting. All right, so just to look at the properties of clinically useful neuromuscular blocking agents. So at this point, we haven't gone into the meat of the matter yet. However, I expect that you have an understanding of these terminologies. So the time of onset the duration of action, the half-life, and the mode of elimination. So we will be going into some of the chemistry associated with the mode of elimination, and we're looking at the structures of these compounds and how they relate to their duration of action and their mode of elimination. So the first compound or drug that we're going to look at is Becamethonium, and basically it has this quaternary ammonium center, well, two of them, and it's joined by 10 methylenes. Okay, so let me play this. So Becamethonium is a simple analog of tuberculin. It's a flexible straight chain molecule and is capable of a large number of conformations. The drug binds strongly to cholinergic receptors and has proved a useful clinical agent, but it suffers from several disadvantages. For example, when it binds initially to nicotinic receptors, it acts as an agonist rather than an antagonist. So it switches on receptors such that the sodium ion channels open up to depolarize muscle cell membranes and cause brief contractions of the muscle. So if we recall in the video that we watched previously, this is the depolarizing muscle relaxant. So it acts as an agonist at first and then behaves as an antagonist afterwards. All right, so this is Becamethonium, which is a depolarizing muscle relaxant. And I mentioned the quaternary ammonium ion, but just to do a brief review of this functional group classification, so we can distinguish between ammonia and primary, secondary, and tertiary amines by the number of carbons attached to the nitrogen. So primary, one carbon, secondary 2 carbons, and tertiary 3 carbons, and these are amines. However, when these amines are protonated, right, we now have an ammonium ion. Okay, and so, well, these two are protonated, so we have our ammonium ion, which only has 3 hydrogens, and we have our primary ammonium ion, which has 3 hydrogens and 1 carbon, and we have our quaternary ammonium ion here. So quaternary suggests 4 carbons here, so no hydrogen attached to that nitrogen. All right, so this leads us now to question 2, which is draw the structure of acetylcholine and compare with the structure of Becamethonium. All right, so pause the video and attempt this question before you move on to the answer. All right, so this is the answer. So we have the color coding here. So we have for acid, we have acetylcholine and we have Becamethonium, right? So for acetylcholine, we have 1 quaternary amine present, so that is in red here. We have our ester functional group present, that's circled in blue here. And we have our 2 methylenes, which are circled here. And just a reminder that methylenes are CH2s. And then we have our Becamethonium, right? It has 2 quaternary amines present. So 1 here and another 1 here. We have no ester group, so ester functional group absent. And we have 10 methylenes present. So you can go ahead and count these methylenes. So those are the structures, and we compared both of them. So this leads us to our follow-up question 2, which is discuss the relationship between the activities of acetylcholine and decamethonium and their structure. So we're talking about structure-activity relationship here. How does their structure relate to their activity? So please discuss that for next class. All right, so our next drug that we'll be looking at, still a depolarizing muscle relaxant. However, this is succinylcholine or suxamethonium. So there are 2 ester groups incorporated into the chain, which I circled in red. So those are the 2 ester group, and they're incorporated in such a way that the distance between the charged nitrogens remains the same as decamethonium. So that should be decamethonium. The ester groups are susceptible to chemical and enzymatic hydrolysis, and once this takes place, the molecule can no longer bridge the 2 binding regions on the receptor and is inactivated. So once the hydrolysis happens, it's now inactive. So the ester groups are also introduced such that suxamethonium mimics 2 acetylcholine molecules linked end to end. So there's a line here, an imaginary line that kind of shows how it's related to acetylcholine. So it's almost like 2 units joined together at the ends. All right, so looking at the mechanism of action of these depolarizing muscle relaxants, particularly suxamethonium. So it physically resembles acetylcholine, which we just established. It therefore acts as acetylcholine receptor agonist, and it's not metabolized locally, meaning at the junction, neuromuscular junction. It doesn't happen there. It's metabolized by pseudocholinesterase in plasma. The depolarization action persists, as we saw in the video and elsewhere, and the continuous end plate depolarization causes muscle relaxation, which we also saw elsewhere. All right, so how are they useful to mankind? So these are 2 clinical uses of these depolarizing muscle relaxants, and I have 2 pictures here which basically illustrates these 2 clinical uses. So it most often used to facilitate intubation or electroconvulsive therapy. And as with most pharmaceuticals, there are associated side effects. So there is muscle twitching, muscle pain, high potassium, which is hyperkalemia, which should make sense to you because based on the mechanism of action, if it causes potassium to be released, then that can result in too much potassium in the system. It causes malignant hyperthermia as well as apnea, which is cessation of breathing. So now we're moving on to non-depolarizing muscle relaxants, which are competitive blockers. So what is their mechanism of action? Well, they compete with acetylcholine at the binding site. They act as competitive antagonists. So if we don't know what that means, we have to check that out. Excessive concentration causing channel blockade. So it actually blocks the channel if you have a lot of it. Act at presynaptic sites. They prevent movement of acetylcholine to release sites, and they do not depolarize the motor end plate, and hence the name non-depolarizing muscle relaxants. All right, so the first one that we're going to speak about is tuberculin chloride, and the structure is here to the right. So let me just calm you down for a second. No, you don't need to know the structure, right? So just relax, take a breath. So curare was first identified in the 17th century when Spanish soldiers in South America found themselves under attack by indigenous people using poisoned arrows. It was discovered that the Indians were using a crude dried extract from a plant called Contradendron tomentosum, which stopped the heart and also caused paralysis. Curare is a mixture of compounds, but the active principle is a cholinergic antagonist that blocks nerve transmissions from nerve to muscle. All right, so this is a picture of the plant itself. So the main application is in the relaxation of abdominal muscles in preparation for surgery. So this allows the surgeon to use lower levels of general anesthetic that would otherwise be required and increase the safety margin for operations. So curare is actually a mixture of compounds, and it was not until 1935 that the active principle, which is tuberculin, was isolated, and we have the structure here. So tuberculin was used clinically as a neuromuscular blocker, but it had an undesirable side effect, as it also acted as an antagonist at the nicotinic receptors of the autonomic nervous system, and that is not good. So the structure, and remember this is from 1935, so at that time the structure presented a problem to the theory of receptor binding, because it is thought that, you know, if we're mimicking acetylcholine here, then it doesn't have an ester. There is no ester to interact with the acetyl binding region. So the positively charged nitrogen on its own is not sufficient, it was thought, for good binding. So why should tuberculin bind to the nicotinic receptor? Why? Well, the answer lies in the fact that the molecule has two positively charged nitrogen atoms. So one is tertiary and the other is quaternary, right? So you should know the difference between the two. So that's where the answer lies, in the fact that we have two positively charged nitrogen atoms. So this is the theory. It is now being proposed that one of the positively charged nitrogens on the tuberculin binds to the anionic binding region of an acetylcholine binding site, while the other binds to a nearby cysteine residue, right? So let's look at this image right here. We have acetylcholine, right? And it's in a receptor. So we have our hydrophobic pockets, right? We have our anionic binding region, so our quaternary ammonium ion, right? It is positively charged and it interacts with our negatively charged or anionic binding region. And of course, there are other interactions, right? So let's look at the above image seen here. Let's look at the key firstly. So this is an oversimplified version of tuberculin, right? So we're looking at the nitrogens at the ends that are positively charged. And then we have these blue squares that represent acetylcholine binding sites, right? And so the theory now is that one of the nitrogen center binds to the acetylcholine binding site, and the other one binds to nearby cysteine residue. So it's nearby, negatively charged, and it binds to that. And that is how they think that that solve the problem of how does it bind without having a ester functional group. This leads us to question 3, which is, how is curare different from tubocurarine? So the answer is, curare is a crude dried extract from a plant called Chondrodendron tomentosum. It is a mixture of compounds which includes tubocurarine. So from that mixture, tubocurarine was isolated. So you might see, if you search some sources, that curare is the common name for Chondrodendron tomentosum. However, for the purposes of this course, we're saying it's a crude dried extract from the plant itself. So our follow-up question 3 says, distinguish between depolarizing and non-depolarizing muscle relaxants. All right, so now we're going to go into long-acting, intermediate, and short-acting non-depolarizing muscle relaxants. So the first is our long-acting pancuronium. So it is a synthetic product, right? It is based on the naturally occurring alkaloid maltodextrin, and the structure is here. And again, it is some indigenous or primitive people who discovered it first. So it's found in arrow poisons used by primitive Africans. Pancuronium bromide, acts on the nicotinic receptor and in the ion channel, inhibiting normal ion fluxes. So what's important to pancuronium in terms of the structure? Well, it has a steroid nucleus. So I circled it here, or 4-ring portion of this moiety. This is the steroid nucleus, and it acts as a spacer between the 2 nitrogen groups, right? The compound also has acyl groups, right, which are circled in red as well, right? And they were also added to introduce 1 or 2 acetylcholine skeletons into the molecule in order to improve affinity for the receptor sites. So remember, the pancuronium is a synthetic product, which means medicinal chemists would have designed and synthesized this compound. And the idea here is that they want to mimic the acetylcholine in some sort of a way, right? So that is why the acetyl groups were added to the steroid nucleus. So pancuronium has a faster onset of action than tubocurarine and do not affect blood pressure. So they are not as rapid in onset as Suxamethonium and have a longer duration of action. So the main advantage is that it has fewer side effects, which is great, and are widely used clinically. So unlike Suxamethonium, it has no agonist activity, and that's why it's non-depolarizing, and act as pure antagonists, so they have no depolarizing effect on target muscle cells. All right, so now we're going to look at the metabolism of pancuronium. So here we see that it undergoes hydrolysis in the liver to the active 3-hydroxy metabolite and the inactive 17-hydroxy and 3,17-dihydroxy metabolites. So here we have, a steroid skeleton that's numbered, and we have position here, and we have position 3 here, and we have position 17 here. So those are the points where the acyl or the ester group is that would be hydrolyzed, right? And so once these bonds are cleaved, then we have 2 hydroxyls. So what this slide is saying is that the 3-hydroxyl is active, meaning it has similar activity to pancuronium itself, the parent molecule, right? So 3-hydroxy, so the entire structure of pancuronium minus this acyl group would be the 3-hydroxy metabolite, and it is active as well. However, the 17-hydroxy, that is the 17 only hydroxy, is inactive, and the 3,17-dihydroxy metabolite is also inactive. So once these undergo their biotransformation, it is excreted primarily in the urine with small amounts in the bile. All right, so this leads us now to question 4, which says draw the structure of pancuronium, and also how many single bonds do you count between the 2 nitrogen groups of pancuronium? All right, so here we see the structure. So I hope you pause the video and drew the structure. And then I have in red my line. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So I count 9 bonds between the nitrogen centers. How much do you count? Do you count 9 as well? So our follow-up question 4 is draw the 3 metabolic products of pancuronium. All right, so this leads us now to another non-depolarizing muscle relaxants. So this is Atracurium, which is intermediate acting. So the design of Atracurium was based on the structures of tuberculin and Suxamethonium. So for the purposes of the next slide, I labeled tuberculin T and Suxamethonium S, right? So just remember that for the next slide. So this is a structure of Atracurium, right? So we're seeing similar structural features. We're seeing our nitrogen centers, we're seeing our esters, and we're seeing some blocking groups. So it's superior to both T and S, as it lacks cardiac side effects, and that's always good, and is rapidly broken down in the blood. That is Atracurium, right? So this rapid breakdown allows the drug to be administered as an intravenous drip. The rapid breakdown is due to a self-destruct mechanism, which we will see shortly. At the slightly alkaline pH of the blood, which is 7.4, so just over 7, so it's slightly alkaline, the molecule can undergo a Huffman elimination. All right, so this is a molecule that we saw a while ago. It's a little bit smaller here, but same molecule nonetheless, right? So just to point out here that we have our ester here and our ester here, which I redrew here. So we have, every time you see COO, this is our ester, right? So let's look at the ester hydrolysis first. So when an ester hydrolyzes, that bond cleaves, right? So looking at our ester here, this bond cleaves. This side becomes a carboxylic acid. This side becomes an alcohol, right? Which I drew here. So this is the alcohol side. This is the carboxylic acid side. So our ester here, right? We have our alcohol side, so this will form an alcohol as we see here. And we have our acid side, so this side becomes an acid, and again we have a quaternary acid here. The other way in which this molecule breaks down is via Hofmann elimination. So we have a cleavage occurring between this carbon and nitrogen bond. The cleavage occurs here, and we end up with a metabolite that is not active, right? So this bond breaks. So we end up with a double bond here and a tertiary amine here as a result of Hofmann elimination. So this is how, that's the rapid breakdown self-destruct mechanism. We can have ester hydrolysis, we can have Hofmann elimination. All right, so after Hofmann elimination, the compound is inactivated because the positive charge on the nitrogen is lost and the molecule is split in two. So the molecule here is splitting two over here, and we have one positive charge here, but the other one is missing, and so the compound is now inactivated after Hofmann elimination. So what are the important features of Atracurium? Well, let's look at it. So the spacer. So a 13 atom chain connects the two quaternary centers. So let's count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So remember, we're not counting the bonds here. We're counting the atoms. So that is an important feature, the space between the nitrogen centers. Another important feature are the blocking groups or units, right? So these cyclic structures at either end of the molecule which block the binding site from acetylcholine. That's very important. And then finally, we have our quaternary centers. These are essential for receptor binding. So as we saw, if we can jog our memory to a few slides ago, where we saw our acetylcholine just sitting nicely in our receptor, and how that quaternary center was interacting with that ionic portion of the receptor, portion of that receptor. Well, clearly that is important for activity. And so we have two of them in this molecule. It's an important feature, essential for receptor binding. If one is lost through Hofmann elimination, as we said before, the binding interaction is too weak and the antagonist leaves the binding site. So after Hofmann, we have one quaternary center. That is not enough. We need two. All right, so what is Hofmann elimination really? So mechanistically, we have our quaternary center here, and we have our electron withdrawing group in the form of a ester, and we have two carbons joining them, right? So this carbon hydrogen bond is acidic and it is broken. So wherever the arrow starts, that's where the electron originates. Where the arrow ends is where it goes, right? So this bond breaks and the pair of electrons end up here to form the double bond. to this double bond seen here. This carbon-nitrogen bond breaks, and that is why the arrow starts on the line, right? So this bond breaks and quenches the positive charge, and that is how we end up with our tertiary amine, and that is the mechanism for the Huffman elimination. So just a little bit more on the Huffman elimination. The ester groups within the spacer chain are crucial to the rapid deactivation process. Ester groups, very important. The Huffman eliminations normally require strong alkaline conditions and high temperatures. However, if we have a good electron withdrawing group and it is present on the carbon that is beta to the quaternary nitrogen center, that causes the reaction to proceed under much milder alkaline conditions that are present in the blood, which is, as we mentioned before, slightly alkaline, pH 7.4. So let's go back to this beta thing. So the carbon that is bonded to the quaternary center is alpha, and the one next to that is beta. So we have an electron withdrawing group in the form of a ester that is bonded to the beta carbon, beta to the quaternary center here, right? So let's repeat what we said. The electron withdrawing ester group increases the acidity of the hydrogen on the beta carbon such that it is not easily lost. Well, let's go back to the previous point, which says the electron withdrawing group present on the carbon that is beta to the quaternary nitrogen center. So we just looked at this. Alpha, beta, electron withdrawing group attached to the beta carbon. So that hydrogen is increasingly acidic because of the electron withdrawing group beside it. So this ester group will pull electrons to itself, right? Once it pulls electrons to itself, it will weaken this carbon-hydrogen bond, right? And once this is weakened, then it has an acidic character. And as we know about acids is that they will liberate hydrogens. And, you know, we're not going to go into the strength of acids, but we know that they can liberate their hydrogens. So this electron withdrawing group pulls electron to itself, weakens this bond, increasing the acidic character of this hydrogen, as we said before. So the Huffman elimination does not occur at an acidic pH, and so the drug is actually stable in solution at a pH of 3 to 4 and can be stored safely in a refrigerator. So just in case you were thinking to yourself, I don't remember much about electron withdrawing group. So let me just do a very brief reminder. So looking here, we have some examples of electron withdrawing group. And, you know, the cyano, the sulfate, the carboxylic acid, the ester as we saw, aldehydes, ketones, nitro group, these are all pulling electrons to themselves and having a deactivating effect. In contrast to our donators or activators, we see, you know, a lot of electrons, you know, nitrogens, oxygens. These are things that push electrons towards something, and these are groups that pull electrons to themselves. So hope that helps. All right, so the final drug that we're going to look at under these non-depolarizing muscle relaxants is mivacurium, and it is short acting. So it is rapidly hydrolyzed and does not undergo Huffman elimination like Atracurium. Right, so as we can see here, similar kind of structure. We have our bulky groups here, cyclic structures. We have our ester, we have our quaternary centers, etc. So similar type of compound. So we're looking again at this table, which says properties of clinically useful neuromuscular blocking agents. So at this point, we have looked at succinylcholine, we've looked at tubercularine, pancuronium, atracurium, and mivacurium, and we should have an idea of their relative duration of action, their half-life, and the chemistry involved in their mode of elimination. Okay? All right. So this leads us to question 5, which says, will the compound shown undergo Huffman elimination? So take a minute, inspect the compound. Will it undergo Huffman elimination? And the second question says, suggest another possible metabolic pathway. So apart from Huffman elimination, what else could happen? So the answer is no. No to Huffman elimination. Why? The electron withdrawing group is not on the carbon that is beta to the quaternary nitrogen center. So we have our nitrogen center here, and we have alpha carbon, beta carbon, gamma carbon, and our ester is attached to the gamma carbon. So no, it will not undergo Huffman elimination. The second question asks about an alternate metabolic pathway. So based on the structure of the compound, we have esters, and so the ester group is capable of hydrolysis. All right, so our follow-up question 5 says, compare and discuss the metabolism of atracurium and mivacurium. All right, so now we're on to the final portion of the lecture, which says drugs affecting autonomic ganglia. So the ganglion stimulating agents, so we're talking about nicotine, lobeline, and dimethylphenylpiperazinium, which is DMMP for short. So of the three, we need to know the structure of nicotine. They stimulate autonomic ganglia preferentially. Only nicotine is used clinically to help people to stop smoking. Otherwise, they are used as experimental tools. So they cause complex peripheral effect associated with generalized stimulation of the autonomic ganglia. All right, so since this is medicinal chemistry, we have to go into some synthesis. And since we're not going to attack any of those other prolific structures, we're going to look at this simple nicotine molecule here, right? So let's look at the first step, right? So let's listen to this. This is the first step. So in the synthesis of nicotine, we're gonna start by going into the mechanism of the first step. So we have our base, which is sodium ethoxide, and we know what bases do. They like protons, particularly acidic protons, and this proton is acidic, so it will abstract this proton. This carbon-hydrogen bond will break and form an enolate ion, which means that we will form the enolate seen here, which is a negatively charged carbon or carbanion, which is now nucleophilic and will now attack our electrophilic center in the form of this carbon, and the electrons will flow in this general direction and kicks out our ethoxide to form our intermediate, which we will see shortly. All right, so that was a little bit fast. So it's the same step. This is our intermediate that was being referred to just now. So we have our ester, we have our amide, which are acidic proton right here. We have our base. So our base abstracts our proton, forms our enolate, attacks our ester, and then we lose the ethoxide to form this intermediate here. So for the rest of the synthesis, we're not going to approach it mechanistically. We're approaching it by looking at the functional groups, and in the presence of acid or a reducing agent, what happens to that functional group. So the first functional group we're looking at is an amide, right? So in the presence of an acid, what happens? What do you see happening? We don't see a ring anymore, right? So clearly this bond is cleaved, and we end up with this side being the carboxylic acid and this side being a secondary amine, right? We have two carbons and a hydrogen. So in the presence of an acid, again, we have hydrolyzation. This bond is hydrolyzed, right? So we have the carboxylic acid and the secondary amine here. From this step to this step, we have loss of the carboxylic acid. So this could happen, just by heating it, we can have decarboxylation occurring. And then from here to here, what do you notice? What is the difference between this compound and this one? Well, should be quite obvious. So we have a carbonyl here, and here we have a secondary alcohol, right? So what is the reaction that would convert a carbonyl to a alcohol, or a ketone to a secondary alcohol? Well, once we see an H in bracket, that suggests a reduction. If it was an O in bracket, that would be oxidation, right? So this is reduction. Going from a carbonyl or a ketone to a secondary alcohol, reduction. So you need to tell me at some point what kind of reducing agents we could use to convert the carbonyl to the secondary alcohol. All right, so the final step is in utilizing acid again. And clearly, if we look at the product and the intermediate here, we can see that cyclization occurs. And if you inspect this, you can inspect this and inspect that. We can see that this is missing from the product, the OH, and the hydrogen here is also missing. So what have we lost? We've lost water, right? And so this is a dehydration reaction in the presence of acid. So where you see this red line here is showing where the cyclization occur. So, possibly this OH was protonated, and then this nitrogen is nucleophilic, attacks our carbon center, we lose water, which is a good leaving group, and we end up with nicotine. So as I said before, we're focusing mechanistically on this first step. The other steps, we're looking at the reaction conditions and the functional group conversions. All right, so the next thing is that we're looking at the effects of ganglionic blocking agents. and we're close to the end now. So the effects are complex, but those for cardiovascular system or CVS and the visceral smooth muscles are the most important. In the CVS, there is a fall in arterial blood pressure and cardiac output, postural hypotension, post-exercise hypertension. In motility of all parts of the gastrointestinal tract and urinary tract is inhibited, and this leads to constipation, urine retention, and sexual dysfunction. So here we're looking at the various internal organs, because remember this is the ganglionic blocking agents, right? And we're looking at, parasympathetic versus sympathetic system. And we're also looking at some of the results of the ganglionic blockade. So what happens once a ganglion is blocked, relaxation of the GI tract, etc., etc. All right, so finally, we're mentioning the mechanism of action for ganglionic blocking agents. So far, we have seen non-depolarizing blockers and depolarization blockers for the other category that we mentioned first, but we didn't see inhibition of acetylcholine release. So that is affected through a magnesium ion or these other compounds. For the non-depolarizing blockers, what should jump out at you is hexamethonium, right? Rings a bell? No? Should ring a bell. We looked at decamethonium. This is hexamethonium, so we don't even need to have the structure here. We should know how many methylenes are in between the ammonium ion center. And of course, we have some other structures. If you look at them, not much in terms of structure activity relationship. The only commonality there is in nitrogen, but they don't look familiar. They are used historically for treatment of hypertension. For the depolarization block, it occurs when the receptors is persistently depolarized by nicotinic agonist or nicotine and results in decreased electrical excitability of postsynaptic cells, right? So we should be familiar with these two already, but at the ganglion, these are the compounds that are effective. So these are the references and my disclaimer, which is all pictures copyright to their respective owners, etc. All right, so please ensure that you answer all questions and you also answer or follow-up questions for discussion. Thanks so much for watching and see you soon.